Good morning, everybody. Thanks for your attention this morning. My background is in academic medicine, but for the past 20 years, I've been involved in startup companies like this one, either as a venture investor or as an operating executive. And like Chris during his talk earlier, what's really gotten me excited about a technology before has been not only is the technology revolutionary, but the technology actually can be applied to a very common condition. And that's what I'm here to talk to you about today. What I'm here to talk to you about today is Alucent Biomedical. We are trying to develop and commercialize a product called the Alucent NVS vessel restoration system with photoactivated linking. It is based on a technology called natural vascular scaffolding. And very simply put, we are pursuing one of the holy grails in vascular medicine, that is stenting open the vessel without the need for a stent. Peripheral artery disease, which is our initial focus as a company, is a very common condition. It afflicts over 200 million people worldwide, over 9 million people here in the US, and as a result, there's a very attractive global market of about $3.25 billion, growing at mid to high single digits. Nevertheless, this condition is afflicted by a very major unmet medical need. If you talk to docs in the space, they're using balloon angioplasty and stenting, a technology that has been around for 50 plus years. And they will tell you that about half the time they have a failure. They have to come back and treat patients again. This failure rate, in my view, is unacceptably high. And too many folks are ending up like this poor guy here with the loss of a limb at the end of the day. It would be remiss of me as the CEO of a company with an exciting technology not to tell you about some of the other applications we have, and I'm going to spend the rest of this presentation talking about peripheral artery disease. But just for a moment, I want to let you know also that this same technology, which I'll describe in a moment, is applicable to many other vascular conditions. In fact, one of them on the list, which is the last one, the progression of AAA disease, is something that has no products in it today. Our technology is based on the observation that in diabetic patients, those patients don't have aneurysmal progression the way normal patients do. That's thought to be due to the glycemic cross-linking of the extracellular matrix structural proteins in those vessels. Our technology is very similar, which I'll describe in a moment, and we can also stabilize that aneurysm and have shown just such that we can do that in standard animal models of aneurysmal disease. But back to PAD, let me tell you a little bit about NVS. NVS therapy, stenting without a stent, is a therapy that's comprised of three components that we integrate together. The first is a novel photoactivatable small molecule. The second is a light fiber that activates that small molecule at the proper wavelength. And the third is an angioplasty balloon. And what we are doing very simply is when we activate the molecule in situ in the vessel wall, that directly links together certain amino acid moieties in the collagen and elastin, the native collagen and elastin in the vessel wall, thereby creating a natural vascular scaffolding effect in the vessel without the need for an implant. The product that we'd actually like to bring to market is this one. You can see on the left-hand side of the screen, this is a fully integrated, very simple to use vascular device. It looks a lot like other products at the cath lab. And it is fully compatible with standard guide wires, standard access techniques. On the right-hand side of the screen, you can see our active molecule, which is coated on the angioplasty balloon. It has a yellowish, orangish color to it. And below that, you can see what it looks like when we activate the integrated light fiber in the device. This simple animation will give you a better idea of how this will work in practice. What we're going to focus in on is a cartoon representation of a lesion in the superficial femoral artery. Once we see that lesion, you'll notice that we cross it with a standard guide wire. We then introduce our device. We blow up the angioplasty balloon. That, of course, accomplishes the angioplasty of the lesion in the vessel and also allows for the diffusion of our proprietary molecule into the vessel wall. We then activate the integrated light fiber in that device. That then causes the instantaneous linking of the collagen and elastin molecules in the vessel wall. And then perhaps the best part of all of this, we then deflate the device and remove it, and we leave nothing behind other than the linked native proteins in the vessel wall. and you are left behind with that natural vascular scaffold. Let me move now to showing you just some of the effects of this. If you blow up a plain balloon in a pig vessel, and the pigs are the standard model for vascular devices, what you see is in this picture. The vessel just snaps right back to its normal, what we call RVD, reference vessel diameter, back to its normal diameter. If, however, you activate it with NVS at the time of that balloon inflation, you can see on the picture on the right what appears to be an angioplasty balloon shape sitting in that vessel. But there's no balloon there at all. The balloon is gone. We have actually remodeled the vessel around that balloon shape. Perhaps more dramatically, we knew that we could also repair arterial dissections with our technology. So Krishna Rocha Singh, who's one of our chief medical advisors, was in the animal lab with us one day, and we said, hey, Krishna, can you take a guide wire and just dig it into the vessel wall and make a dissection for us? And then let us see if we can actually repair it. Well, Krishna got a little overly exuberant that day, and what he actually did, that you can see in this picture here, is he actually perforated the vessel. And you can see the contrast leaking out of it. You can appreciate that the vessel's all spasmed down from the injury. We said, all right, well, let's go ahead and treat this anyway. And you can see the effects on the right. And here you'll notice that not only is the perforation sealed closed, but you see that nice open balloon shape despite the fact of all of the vascular spasm on either side of it. There were two of our other scientific advisors in the room that day, and I watched their eyes kind of widen when we showed these pictures, and that's when I knew I was in the right place. In order to bring a product like this to market, we have to have an executive team that's skilled not only in the typical medical device arts, but also in pharmaceutical development. I'm very proud of the team we brought together here. I'm not going to go through all these bios, but you can see that we have kind of the usual suspects, as I call them, from device companies, folks that have led R&D teams repeatedly in cardiovascular device companies and done clinical development in those companies, combined with folks who have spent 20, 25 years in the pharmaceutical industry doing vascular biology, cardiovascular pharmacology, small molecule development. In addition to that, we've managed to attract some of the leading lights in the space. If you've been in and around the vascular intervention realm, you'll know some or all of these names. Elzer Edelman is one of the godfathers of drug delivery to the vasculature. Gary, Bill, Craig are always up on the podium speaking about new technologies or PIs of major multinational trials. Craig actually is a clinical site for us, and Bill just got finished presenting our technology at Link, one of the major conferences. Where we are today is that we've spent the last 2.5 years performing extensive preclinical proof of concept. We've treated over 70 large animals. We've actually done chronic animal studies in the one disease model that does exist in transgenic pigs and shown effective results that actually last. In addition to that, we've done multiple perfused human cadaver experiments, and this was critically important to show that we could actually get our therapy in the actual disease. Lastly, we've spent a lot of time doing scientific background information, basic science, demonstrating the tissue effect, the collagen density effects of what we actually have done with our molecule and our therapy. We are imminently enrolling the first patient in our US phase 1 study that is occurring. We actually had our first site initiation visit last week. And our plan after that is to file an IDE to move into a larger 75 to 100 patient study with our commercial product next year. And that's actually the study that we're looking to raise money around. We're planning a $30 million financing, which will be our first externally led round of financing. And the primary goal there is to complete this 75 to 100 patient study with our commercial product to show robust proof of safety and efficacy. We have a single owner today, which is Avera Health. It's a Midwestern hospital system. This technology was spun out of their research institute, and they have been very generous with us. They put over $20 million of capital to work in the company to date, and will be a significant investor in the next round as well. Those of you who maybe have not paid attention to the peripheral artery disease market recently might not know why it makes sense to pay any attention to it. That unmet need that I showed you earlier on has resulted in a bunch of companies being formed with new technologies. Over the last dozen years, many of those companies have been Very exciting investment returns for people who have put money to work, and this is a list of some of those companies. For us, we have a very simple yet compelling investment thesis. Firstly, the market-leading companies are out there treating patients, marketing products, largely balloons and stents with outdated technology. Despite what the marketing arms spend a lot of money on, there's relatively poor differentiation of those products company to company. And we know that there are major clinical outcome limitations. Lastly, there are now new safety questions around the use of one of those old technologies, paclitaxel, in the lower extremities. Nevertheless, these outdated technologies are significant profitability drivers for those incumbent companies. And here we come along with technology, NVS, that has the opportunity not only to dramatically improve the therapy in this space, but also to be significantly differentiated. And I believe that the existing sales forces will feel they need to have this in their bag once we show that this works clinically. Thank you for your attention this morning.